All right, good afternoon, everyone. I'm Lusia Mhuulu, as mentioned, and I'm going to present our research on evaluation on targeted next-generation sequencing, and I am from the University of Namibia under the Group of Research in Infectious Disease. So I have no conflict of interest to disclose. As we all know, TB is a major global health burden, with drug resistance as a major obstacle which in ending TB, which means there's a need to have a rapid and accurate detection of drug resistance for effective treatment of TB. And so from now, the WHO TB, so we have in Namibia, we have over 14,000 estimates, and for drug resistance, we have about 630 estimates, as Elga mentioned, but only half of those drug resistance are being detected or with a confirmed drug resistance. And so, which means there's a need to upscale the drug resistance, or basically diagnosing drug resistance in Namibia. So we have for new drugs and some of the second line, so there's no phenotypic DST done in the country, although all these drugs are available for treatment. And for targeted next generation sequencing is increasingly adopted due to reduced turnaround times and high diagnostic accuracies. And for Namibia, so have adopted the targeted next generation sequences as it appears in our diagnostic algorithm. So for this study, we look at basically assessing the diagnostic performance now of targeted next generation sequencing using clinical isolates with various resistant profile. So for this, we isolated DNA from 119 Mycobacterium tuberculosis isolates using now the duplex assay. So we isolated the DNA and then do the PCR using the duplex assays, and then do a library preparations, and then analyze using the cloud-based analysis, which gives us now the comprehensive drug resistance for about 15 anti-TB drugs. And then the same DNA, or basically an aliquot, was used to do a whole genome sequencing to compare now our TNGS basically to whole genome sequencing, which was done in the Research Center Bosel. So the targeted next generation sequencing was done here in Namibia at the University of Namibia laboratory. So for the results, when we compared the whole genome sequencing profiles now, the targeted next generation sequencing to whole genome sequencing, so there was a concordance of over 90% for all the antibiotics. And also, so when we look at the specificities and sensitivities of the two, basically of targeted next generation sequencing, it was mainly above 80% for most of the drugs, and the low, or basically the discrepancy was mainly due to the low frequency mutations, and then for variance in interpretations for ethionamides mutation between the two methods that was used. So overall, so we had a high concordance between the two methods, which means targeted next generation sequencing shows a strong potential as a rapid and scalable TB drug for resistant detection. And so, which means also targeted next generation sequencing support personalized treatment strategies in TB care. And we can, so also targeted next generation sequencing could be used or serve as scalable alternative to traditional phenotypic DSTs, particularly in the resources limited settings. Yeah, with that, I thank you